Hello, my name is Rachel Krieger. The date is 5/4/23, and I'll be presenting today on Wilson's disease. Introduction. Wilson's disease is an inherited disorder which leads to an excess amount of copper accumulated in the organs. Hisari, 2019. It results in a lack of copper removing, or it's a result of a lack of copper removing proteins that are present in the body. It is started when there's a mutation in the ATP7B gene. This generates a copper transporting ATPase2 protein normally that is responsible for transporting copper from the liver to other parts of the body. And this also eliminates the excess amount of copper in the body. This mutation of the ATP7B gene also affects the PRNP gene, which is responsible for providing instructions for making a prion protein, which is a protein found mainly in the brain. Specifically affects the 129 position of the prion protein. This means that that protein is active in the brain and other organs and is responsible for the transportation of copper. Therefore, when there is also an effect on that gene, then the function to remove copper from the brain and other organs is depleted. There's also an issue of excessive amounts of copper in the mitochondria. This leads to a decrease in the production of ATP anaerobic respiration, which aids in most of human function. The signs and symptoms of Wilson's disease includes liver failure, neurological damage, and psychiatric issues. Copper depositing in the cornea and front surface of the eye, which manifests as a Kayser-Fleischer ring that most physicians test for when diagnosing Wilson's disease. There's also abnormal eye movements and limited eye dexterity in many cases. The excess of copper in the mitochondria of all the cells, especially the liver cells, leads to fatigue, shakiness, or uncoordination in the muscles. Swollen blood vessels, abdominal pain, and yellow eyes and skin also lead up to this. The diagnostic methods that are involved in Wilson's disease begins with the preliminary testings. This begins when there are signs of the Kayser-Fleischer ring, which is tested with the slit lamp test on the eye. There's also testing for the blood and urine once there is a yellowing of the skin, which we test for excessive copper levels, which can show that there's an excess amount of copper and urine in the systemic body, like the circulatory system and the digestive system. There's also a liver biopsy that can be done that can test the actual liver cells and see if they are failing due to excess copper. However, all these preliminary tests only show that there's an issue with liver and an issue with copper removal from the body. It does not specifically mean that the person is positive for Wilson's disease. And that was stated by Karad Mayi in 2010. Only genetic testing can positively conclude that the patient presented with liver issues and copper removal issues can be tested for Wilson's disease and show a positive test for that. That would involve taking some blood samples and putting it into a purple top EDTA tube. This is stated in Nguyen's 2018 presentation. And then they're able to analyze the presence of the mutation of the ATP7B gene, which is highly correlated to Wilson's disease. It is able to test for specific mutations, such as if a deletion happened or a duplication of the genome, for example, and this test is required by a specialist to order and perform. The influence that Wilson's disease has on the pathways and cellular function starts with the ATP7 gene mutation causing the copper transporting ATPase2 protein enzyme to become non-functioning. This leads to copper not being transported properly out of the liver and other parts of the body as far as 2005. Again, the PRNP gene is also affected, which is a precursor to the prion protein, which as a result of this mutation is non-functioning. As a result, copper is not taken out of the brain and other central nervous system tissues such as the eyes and even possibly the spinal column, which is why we see tremors and other muscle issues, as well as the mitochondrial excess of copper. Methionine instead of valine is at the position in the 129 of the prion protein, and that can also be tested for and indicate what is wrong. This leads to increased neurological disorders and tremors, as said by Espardo 2005. And as you can see, there's a diagram over here listing all the issues with Wilson's disease and how this really can become a whole systemic issue instead of just a simple deletion or insertion of a mutated gene. The cellular functions that are influenced because of this excessive copper is mainly seen in liver cells and muscle cells. In liver cells, this leads to too much copper in the liver and eventually organ failure from the liver. The cells in the liver and the muscles try to rid themselves of the copper, but then this copper is just leaked into the extracellular matrix, leading to the cell death and the muscle and other functioning issues of the organs. There's also a cell cycle checkpoint failure that we see due to this mutation is during the chromosome segregation, and there's a failure to check the viability of the cell, which could lead to misfunctioning cells that do not do what they need to do. For example, taking copper out of the body and ridding it of that in a proper manner. Burkhead 2011. The genetic factors that are involved in this is a mutation that, again, has happened in the ATP7B gene. This codes for the copper transporting ATPase2 which is responsible for transferring copper from the liver to other parts of the body, ARCA 2022, is an autosomal recessive genetic disorder, meaning that is needing both of the recessive alleles to show presentations of clinical issues associated with Wilson's disease. However, if someone has just one of these recessive alleles that codes for the Wilson disease, then they are classified just as a carrier. There, however, has been not a sex link to it, but there has been more acute liver failure because of Wilson disease in more women than men. And that was found out by Abdel Ghaffar, 2008. Again, it's a homozygous or a compound heterozygous mutation that is a frame shift and a nonsense mutation. A nonsense mutation occurs when a normal sense codon is changed to a chain terminating stop codon, which leads to an unforeign protein, which is the reason that the ATPase2 copper transporting protein is unfunctioning. And that is stated by Hura, 2021. The epigenetic factors aren't really much. It is an autosomal recessive disorder, so there's no clinical presentation in those carriers as a mutated allele. There is heterogeneity in age of onset. It is hard to pinpoint, so that means that it's hard to pinpoint diagnosis of disease. Physicians have a hard time controlling the symptoms, age of onset, and genetic variations, and the resulting clinical consequence of such variations. And it's hard to take all those together and make sure that it is Wilson's disease as compared to another disorder that could affect copper depositing in excess amounts. There was a case reported of a pseudo-dominant inheritance where many of the same family were all affected with mutation, but this was just a coincidence and it was not some anomaly that was an unknown mutation or a novel mutation. And that was reported by Chang in 2017. The generational impact starts with the fact that there is an autosomal recessive trait in Wilson's disease. It does have that characteristic. That means that the person who does have clinical signs of Wilson's disease, including the Kaiser-Fisher ring and everything else, they're required to have both recessive alleles in order to phenotypically display these signs of Wilson's disease, meaning that it is monogenic in nature. If you were testing positive, for example, if there's one person that has tested positive for Wilson's disease, most physicians do encourage their other family members to get tested genetically so they can be properly treated if they are below the age of where most symptoms come about. So most symptoms come about in late teenage to 30 years of someone's life. So there is a large gap of when this does come. That's why physicians are a little unable to always immediately diagnose someone without proper genetic testing. And it is highly improbable that two generations with recessive alleles in both, as stated by Todorov 2016. However, it is good to test who has the recessive allele so that maybe, and who is a carrier so that they can be aware of the issues with having children and basically creating and also giving them the carrier for the allele. And the treatment that's involved with this is pretty simple, but first we do need to begin with proper diagnosis. As I said before, the preliminary testing with just the blood and even the Kaiser-Fisher ring isn't always 100% accurate in diagnosing specifically Wilson's disease. It could have led to another thing or be a result of another disorder. So we want to be very accurate in our genetic testing. Then once a diagnosis has been confirmed that it is Wilson disease, a treatment plan can be made once that is established, and once it's established how far into this disorder and how damaged the organs are. So the main concern with Wilson disease is an excess of copper, which leads to the copper toxicity in both the blood and the liver and the urine. And as a result, we need to reduce the amount of copper consumed in a diet, and we can find ways to rid the body of the excess copper. And once that is done, we kind of just focus on maintenance of keeping that copper level low in the body. The easiest way that doctors have found to quickly rid the body of copper levels is through albumin dialysis. And there are other therapy options once the doctor has approved and made aware everyone of what the situation is in regards to age, other factors, and everything else that he thinks is necessary or they think is necessary. Usually the patients with excess copper are given chelating agents, which work by binding to copper into tissues and promoting organ release of copper into the bloodstream so that the body can filter that copper out through the urine. There are two common chelating agents that are used. One is D-penicillamine and the other one is trientine, an assay by Wiskerchen 2019. There are also zinc salts that are used, which Hefter 2022 was able to report a positive result from the zinc salts as well. If the liver biopsy that was done previously shows that the tissue is highly damaged and there might not be really a chance of saving that liver, and it could possibly spread to other organs and cause them to basically die off and misfunction, then a liver transplant might be advised. Again, there is dialysis, there's specifically albumin dialysis of the blood to remove the copper, and Kaskakova 2016 was able to report a successful removal of copper from that dialysis. And after once all the copper levels are at basically homeostasis where we want them to be in normal range of a human, then it's basically just about management up till then, and we're going to reduce the amount of copper that we consume. Since copper is not really abundantly made in the body independently, it is fairly manageable. Some novel researches that have been going on include new diagnostic methods called the Lipstick scoring system. This uses brain magnetic resonance imaging features on top of family history that creates a specific score. If one receives a score of 4 or above, then they are able to accurately diagnose a Wilson disease in this patient. And as you can see, this is the effect of Wilson disease on the brain because of that prion disorder, as well as copper just not really being removed from the brain itself. As you can see, there's deterioration over the passage of time, and on the right, you can see the full effect of Wilson's disease if it goes untreated. With gene therapy, there is a lot of promising research being done, especially with the adeno-associated virus, which is AAV, and they are vectors that we can use alongside liver-specific promoters to kind of change how the proteins that are mutated can be expressed and translated different. The AAV is a virus with protein shells that is surrounding and protecting a small single strand of DNA genome of approximately 4.8 kilobases, and that is stated by Wesker in 2019 in their research. The virus does not, however, cause disease in humans, which means it is very advantageous in treatment of cell functionality issues, and in this case, gene therapy. The administration of the AAV is usually best for when there's not liver or other organ failure, and it is able to partially or fully reverse the disease. And if there is already liver damage, then a larger dose of AAV is required for possibly successful treatment. The second method, when there is a liver failure already, has not shown to be as successful as before the onset of liver failure. However, it still has some promise. It is in the trial stages, and ESA 2023 also was able to report on that. There are other researches being done in how to manage and control copper levels best in humans. There is studying and research being done on the use of anion exchange chromatography that is coupled to triple, quadruple, inductively coupled plasma mass spectroscopy. This is promising, but is still in the preliminary stages of rat testing. There's also use of methanobactin, which is a peptide produced by Proteobacterium methylocyanus trichosporium. And this is used by physicians to remove excess copper from the mitochondria specifically, so we can decrease liver histopathology and prevent liver failure. So this helps with the tremors as well as your liver function, and hopefully not allowing the liver to get to a point where it is in the organ failure stage. Again, the mitochondrial dysfunction is an important characteristic of Wilson disease, stated by ESA 2023. The lifetime outlook for Wilson disease patients are pretty manageable. Once the copper levels are stabilized, all efforts are put towards maintaining those healthy levels, and it is fairly easy by just avoiding copper in their diet. And again, the copper levels in the body usually aren't that high anyways, because this is not a natural occurring substance that the body can make independently. They also, because of some potential liver damage that has happened either in the past or could in the future, they are usually advised to stay away from heavy drug and alcohol use. The sitting supplements are able to aid the body in reducing the amount of copper that it could potentially absorb by binding to those copper elements as well. We can also use vitamin B6 that also works similarly. And then patients are also highly encouraged to get vaccinated for hepatitis A and B, and this is to ensure there is no other potential damage that can be done to their liver, similarly with the alcohol and the drug use, and as stated by Hefter in 2022. Patients are also given medicine to treat their muscle tremors and stiffness if the muscles and their nerves are beyond repair, and right now all you can treat is those. These are my references, and thank you.